So just moving on, our second speaker is John Brestoff. He's a PI at Washington University of St. Louis. So John completed his PhD and MD at UPenn, and there he was studying how innate lymphoid cells regulate thermogenic VEGF fat and limit the development of obesity. This work was published in Nature in 2015, and after that, he moved on to Washington University in St. Louis, where he's right now. And at WashU, he was studying what I believe is the work he's talking about today, and that's going to be intercellular mitochondria transfer to macrophages. So this work, if I'm correct, was published earlier last year in Cell Metabolism. And yes, I'm looking forward to your talk.
Sorry about that. I wasn't able to unmute myself while I was in presentation mode. First of all, thank you so much for the introduction, and thank you to the organizers for the invitation to share the work with you. So today, I'll tell you about the story we published last year on mitochondria transfer in the adipose tissue, and I hope to give you a little bit of a teaser about another paper on this topic that we have that we've been working on. So I do have some patents related to obesity and mitochondria transfer that are pending, but they're not really related to the content of this particular talk. So my lab is really focused on understanding how the immune system interacts with metabolic organs to control their function. We're really interested ultimately in understanding how the immune system regulates physiologic adaptation to nutrient stress and to metabolic disease pathogenesis. So the lab studies this as well, and about half of the lab is also really focused on mitochondria transfer. We're interested in this problem in part because obesity is a major public health problem worldwide. In the United States, where this disease is particularly prevalent, it's estimated that at least 42% of adults are clinically obese. This is problematic for two reasons. One is that it's an independent risk factor for developing many other diseases, including type 2 diabetes. And the other is that it costs a lot. In the US alone, it costs about $200 billion per year in our healthcare system, at least before COVID. So ultimately, obesity is caused by chronic positive energy balance, where energy intake exceeds energy expenditure. And this leads to storage of free fatty acids in triglycerides, in adipocytes, and sometimes ectopic cell types. And what's really interesting to me about this is that in the last 10 or 11 years, we've started to appreciate that the immune system actually can regulate this energy balance equation. And this is what my lab is really interested in studying. So white adipose tissue is traditionally thought of as being an endocrine organ and that produces hormones to regulate a bunch of different physiologic phenomena, and also an energy storage organ. But what surprises people often is that a healthy adipose tissue contains a lot of immune cells that tend to be skewed towards the type 2 immune system. They're enriched in, especially in eosinophils and group 2 innate lymphoid cells, which I studied during my PhD with David Artis. And these two cells work together to coordinate the function of macrophages. They get a little bit of help from T cells. And ultimately, these macrophages have emerged to be central regulators of metabolic homeostasis. They have been ascribed many different functions, and there are several different subsets of macrophages in adipose tissue. But some of the most important ones are that these macrophages have been shown to regulate and sustain insulin sensitivity, at least in adipocytes. They also regulate lipolysis and the release of free fatty acids into circulation. And they have been shown to increase energy expenditure by eliciting beige fat and brown fat activity. In the context of obesity, there are some adipocytes that undergo cell death, and this leads to the release of all of their cellular contents into the interstitial space. This is a very pro-inflammatory process, and as a result, there's a recruitment of macrophages that are very highly phagocytic, where it's thought that their job is to come in, phagocytose, and eliminate all of the debris that's been released into the tissue. These macrophages actually they surround this dying adipocyte, and it looks like a crown sitting on top of a head. And so this morphologic structure has gotten the name the crown-like structure. This process is associated with elaboration of type 1 cytokines, especially TNF alpha and interferon gamma. There are also some metabolites like palmitate that get released that are also pro-inflammatory, and there's even LPS that can get to the adipose tissue from a leaky gut. And these factors all work together to promote inflammation in macrophages. And this process has been linked to impairment of insulin sensitivity in the adipose tissue, as well as triglyceride storage and impaired energy homeostasis. And so these this macrophage polarization scheme has been linked to obesity pathogenesis. So we have learned quite a bit about how macrophages are regulated in the adipose tissue, but we still don't have a complete understanding of all of the mechanisms that control macrophages in fat. So when I was a resident here in pathology, I had some time to really think about and get creative with science. And I came across some papers that really just kind of blew me away. The first actually is one of the first papers on mitochondria transfer from Darwin Prokop's group, which showed that there are mitochondria deficient Rho 0 cells that can capture purified mitochondria from supernatants and use those mitochondria to support aerobic respiration and cell division. There have been a number of papers that have reproduced that finding in vitro, and a few papers that have shown that mitochondria transfer can occur in vivo, including one from Eng Lo's group from Kazu Hayakawa, showing that astrocytes can actually transfer mitochondria to ischemic neurons in the setting of ischemic stroke. And so there are actually 3 different companies that are trying to leverage the mitochondria transplantation technology for therapeutic purposes, which are listed here. I have no involvement with any of them, but they've all seem to be doing pretty well. So when I first embarked upon this project, there was really very little known about mitochondria transfer and its mechanisms. And we wanted to know whether or not this process was involved in regulating macrophages in the adipose tissue. So to begin investigating this, we started with a very basic question, which is whether or not any immune cell populations in adipose tissue could acquire mitochondria from another cell type in vivo. So to accomplish this, we also used the MT-dendra2 construct, and we had a mouse where the MT-dendra2 was expressed in every cell type. We also had wild type mice, and both of these are on a CD45.2 background. We then lethally irradiated these mice and then rescued them with a bone marrow transplant of CD45.1 congenitally disparate wild type bone marrow. This is just a marker that allows us to determine a donor versus a recipient derived cell. We then let them engraft, and this for 12 weeks, and this allowed us to generate mice where we had global mitochondrial reporter mice that contain a wild type immune system. So the donor cells cannot make the D2 construct. We then isolated the white adipose tissue and did flow cytometry. And for those of you who don't look at flow plots all the time, I thought I'd just take a second to kind of orient you. So so basically this is just an XY plot. And the signal intensity for the x-axis is CD45.2, and for the y-axis is CD45.1. And each dot that's shown on this plot is an individual cell that's plotted just based off of how intense the expression level is for these different markers. And then there's a contouring function like a topographical map looking down on a mountain range to show you just how dense the cell population is. And so these cells here are radio-resistant host cells. And these cells are wild type donor cells. So these donor cells cannot make D2. And so we drilled down into this population of cells, and we found that about a third of the immune cells that are present in the white adipose tissue of the recipients that express D2 in their host cells were positive for the D2 signal, suggesting that a substantial proportion of the immune cells in the white adipose tissue were at least able to physically interact with mitochondria derived from a host cell. When you drill down into this population of cells, we found that the majority of these cells are actually macrophages. About 2/3 of them are sort of conventionally defined macrophages, and then there's some other cell types too, but they're mostly myeloid cells and a CD11C cell population that's probably, based off of the literature, probably also another macrophage subset. So one of the advantages of flow is that it allows you to do very high throughput experiments, but the disadvantage is you get no spatial resolution. So you don't know whether or not these D2 mitochondria are actually in the cell, or whether they're just stuck to the surface, or if there's just some diffuse cytoplasmic signal. So to address that limitation, we turned to an imaging approach with intravital to photon microscopy. So in this experiment, we isolated wild type macrophages from the white adipose tissue, and then we labeled them with a red dye called CMTMR. It's a cytotracker dye, and this is frequently used in the immunology community to track adoptively transferred cells in the recipient mouse. And so we just did an IP injection, and it's known that these macrophages can actually somehow relocate back to the adipose tissue. And so we injected them into the global mitochondria reporter mouse as a host. And then the next day, under general anesthesia, we opened up the peritoneal cavity, placed the objective right on top of the adipose tissue. And then with this technology, you can image about 35 to 40 microns deep into the tissue. And you can do time-lapse imaging, you can do 3D positional reconstructions. And here is what it looks like. So these black circles are actually the lipid droplet in the adipocyte. This is a red wild-type donor macrophage intercalated, and then all these green puncta are host mitochondria. And what we found was that in almost every macrophage that we could identify, that there were these yellow puncta, which we interpret to be internalized mitochondria. And actually, after acquisition, we were amazed to find actually this, which is in the mitochondrial capture event, where you can see this mitochondria being taken up and then worked on with fusion and fission. And now we're just walking the 3D reconstruction through in a still image and now through a moving image, so that you can see that these yellow puncta are really in the cell. They're not sitting above or below. So this was a very important experiment for us because it showed that mitochondrial transfer is occurring natively in the tissue, that this process occurs very frequently, and that, that this is a dynamically regulated process. And one thing I actually normally don't point out, but I will because of Michel's talk, is that there's actually some evidence that some of these mitochondria actually get exported on the other side. So it almost appears that macrophages can sometimes take up mitochondria and then possibly package them and deliver them to another location. Okay, so we thought that given the anatomy, that adipocytes would probably be a very likely source of mitochondria. So to macrophages. So we to test this, we developed a mitofat, what we call a mitofat mouse. It's actually the Dendritus stopflox mouse that's crossed to adiponectin-3. And this allows you to generate an adipocyte-specific mitochondrial reporter mouse, where only mature adipocytes express Dendritus. It's a very good CRE that we're using for this system. And then we isolated the epididymal white adipose tissue and took the stromal vascular fraction for flow cytometric analysis of the D2 signal in macrophages. And what we found was that when we look in the macrophage compartment in the control mice, there are very few events where there are D2 positive macrophages, but in the mitofat mouse, about 40% of the macrophages were D2 positive. And we confirmed using intravital to photon microscopy that these macrophages really are taking up mitochondria from adipocytes in vivo in the tissue in situ. And this mouse has turned out to be very important for us because we can do a lot of really cool experiments like this one, where we sort purified the macrophages that have or haven't taken up mitochondria from adipocytes. And then we did RNA sequencing. And you can see in this PCA plot that these two macrophage subpopulations, which came from the same tissue and were processed identically, are transcriptionally distinct from each other. We did some other comparison showing that there are some metabolic differences between these two cell populations. And there's still an open question about whether or not mitochondria transfer is actually conferring these transcriptional changes and metabolic alterations, or whether or not these are just two different populations of cells with different efficiencies for mitochondrial capture. So we wanted to be able to genetically manipulate mitochondria transfer to study its physiologic function. And at the time, there was really very little knowledge about what factors could regulate this process. So we wanted to know whether or not there were any receptors for mitochondria or any attachment factors that we could genetically manipulate to disrupt this process from occurring. So to do this, we performed a genome-wide CRISPR knockout screen. We did this in collaboration with Craig Weiland, who was in Skip Virgin's lab at the time, and John Dench at the Broad Institute. And what we did was we obtained BV2 cells, which are actually microglia derived, but in vitro, they behave a lot like a macrophage. They were transduced with Cas9, and then we introduced the Brie CRISPR library. So this library has 4 CRISPR guides for each of 20,000 genes. And then we exposed this pool of cells to D2 mitochondria. And we optimized it so that basically every cell would take up mitochondria. And then we sort purified the negative cells. The idea being that these cells contain a CRISPR guide that is targeting a gene that's essential for a cell to take up mitochondria. So we sequenced for the CRISPR guides relative to the input. And we had 23 genes that had a false discovery rate that was less than 0.05. It's actually a very stringent cutoff. Most labs use a false discovery rate of around 0.2, 0.25. But we had such a good screen that we thought that this was actually a reasonable cutoff point. So I'd actually never heard of any of these genes before. So I put them into the GO-Panther pathway analysis and found that there was really marked enrichment for the heparan sulfate proteoglycan biosynthesis pathway. And it turns out that of the 23 genes that we identified, 13 of them are involved in heparan sulfate biosynthesis. And in a completely unbiased fashion, every single gene in the heparan sulfate biosynthetic cascade was identified by this screen, including all the enzymes involved in the formation of the GAG linker chain, the exit complexes that are responsible for elongation of the heparan polymer, and then the sulfation enzymes involved for some of the sulfation events, and also some of the substrate synthesis enzymes. Still blows me away. And so we hypothesized, well, I should also mention that the exit1 gene was our #1 hit from the screen, and this gene's actually been linked to some metabolic alterations in mice and humans, and exit2 is actually is a SNP linked to human type 2 diabetes. Okay, so the screen led us to hypothesize that maybe heparan sulfates are involved in mitochondria transfer. So to test this, we did a bunch of things, some of which I'll show you. First of all, we made clonally selected cell lines for every one of those genes and showed that all of them have impaired capture of mitochondria, but not latex beads. Then we treated the BV2 cells with heparinase. So this will enzymatically remove all the heparan sulfates on the surface of the macrophage or of the BV2 cells. And we showed that that is sufficient to inhibit mitochondrial uptake in vitro. Then we also pre-treated the mitochondria with heparan sulfate proteoglycans, and this was sufficient to competitively inhibit mitochondrial uptake in vitro as well. So we had genetic, enzymatic, and also pharmacologic data suggesting that at least in vitro, this pathway is operative in capturing mitochondria by macrophages. So to test whether or not this occurs in vivo, we treated our mitofat mice with heparin. Heparin is an anticoagulant that we use clinically all the time. And it turns out it's just very highly sulfated heparan sulfates. And so we treated mice daily for 7 days, and we found that this was actually sufficient to inhibit mitochondrial transfer from adipocytes to macrophages in vivo. We never get a complete elimination of mitochondrial transfer. There are probably multiple mechanisms by which mitochondria are transferred, and we're just interrupting one of the mechanisms. So I told you before that in obese adipose tissue that there are macrophages that get recruited to around these dead adipocytes to eliminate the debris that's released into the tissue. And this is a really nice IF image from Kerry Lemang's lab showing that these macrophages are actually engulfing material released from the adipocyte. And it's well documented that the pro-inflammatory signals like interferon gamma, LPS, and TNF increase the phagocytic capacity of these macrophages to facilitate that clearance process. And so we hypothesized that there must be an increase in mitochondrial transfer from adipocytes to macrophages in the context of obesity. So to test that, we took our mitofat mice and we put them on a high-fat diet for 12 weeks compared to chow, and then looked for D2 signal in the macrophages. And we actually found the total opposite. We found that actually the degree of mitochondrial transfer was reduced from about 40% in a lean state to 10% in a setting of a high-fat diet. This was associated with reduced heparan sulfate levels on the surface of the macrophage. Then we used some co-culture experiments to figure out that it's actually not due to impaired release of the mitochondria from the adipocytes, but rather impaired capture of the mitochondria by the macrophages. So we wanted to know whether or not this process is involved in obesity pathogenesis. And to accomplish that, we actually were lucky enough to be able to get exit 1 flox mice. We crossed these mice to the lysine cre, so this will delete heparan sulfate synthesis in all myeloid cells, including macrophages. And then we confirmed by flow cytometry that we successfully reduced heparan sulfate levels in the macrophages. We never get really a complete loss of heparan sulfates, but it is a marked reduction in heparan sulfates. They can probably get a little from drogocytosis from other cells. And then in an ex vivo assay, we found that these cells were also less efficient at taking up mitochondria from adipocytes. So it looked like we were able to impair mitochondrial transfer in vivo with this mouse. One of the things we noticed was that they were a little bit bigger. And so we put these mice into our body composition analyzer, and we found that sure enough, that the exit 1 conditional knockouts had increased fat mass, about twice as much fat as the flox litter-mate controls. So there's really only one way that can happen, which is that there is a disruption in energy homeostasis. There's chronic positive energy balance that's leading to weight gain. So my lab has a dedicated metabolic cage facility. We have that allows us to really interrogate all of the parameters that contribute to energy homeostasis. So this has got, all the racks are actually in an enclosure here that allows us to control temperature and light. We have 16 metabolic cages that are arrayed in this enclosure. And we put one mouse at a time in each cage, and it allows us to measure energy expenditure, food intake, activity levels, and we can basically determine all the parameters that contribute to energy in and energy out. Here are probably the most important piece of data from this experiment. The y-axis shows energy expenditure, which is expressed as heat. And the y-axis is Zeitgeber time. So this means time giver, and it's actually the entrained signal, which is actually the light. So here you can see it's ZT0, this is 6 a.m. when the lights come on. ZT12 is 6 p.m. when the lights go off, and then their mice are active during the dark phase, and then ZT24 is the completion of the light dark cycle. And what you can appreciate is that the conditional knockouts, which are shown in red, they have lower energy expenditure in both the light phase and in the dark phase. But food intake and activity levels were unaltered. So this suggests that there's somehow these mice have an impairment in energy expenditure. We don't really know how yet or why, but that's definitely the case. These data are actually combined data from 4 independent cohorts with litter-mate males, all age matched, from I believe about 20 mice per group. So given those data, we hypothesized that these mice would probably be more susceptible to diet-induced obesity. And so we put exit 1 flox and lysine cre conditional knockout littermates on a high-fat diet for 9 weeks. And we found, sure enough, that the exit 1 conditional knockouts gained significantly more weight on a high-fat diet than their flox littermate controls. They gained more epididymal white adipose tissue. They gained more inguinal white adipose tissue, which is sometimes referred to as beige fat. And they also, but they did not have a difference in brown fat mass. This was correlated with a couple of different obesity-associated metabolic disruptions, such as impaired glucose tolerance, although that is sort of a mild phenotype. So we published this work last year in Cell Metabolism and showed that in the lean state that adipocytes frequently transfer their mitochondria to macrophages through a mechanism that is at least partially mediated through heparan sulfates. In the context of obesity, there are pro-inflammatory factors like interferon gamma and LPS that actually shut down heparan sulfate synthesis machinery. And we believe that this is the reason why there's impaired mitochondrial transfer from adipocytes to macrophages in obese adipose tissue. We think that that mitochondrial transfer is actually metabolically beneficial and that it's intact in healthy mice. But if you genetically disrupt mitochondrial transfer from occurring, that there's an increased susceptibility to obesity and weight gain. So a few months after we published that paper, Claire Crew, who's actually on faculty at WashU now, when she was in Phil Scherer's lab, published this paper also in Cell Metabolism, showing that a high-fat diet can stimulate adipocytes to release their mitochondria into the circulation, and they can get to the heart, where they can lead to metabolic preconditioning to support metabolic adaptation to a high-fat diet and the heart. And so we're actually collaborating with Claire Crew now, and we have a paper that, that is under review where we have basically figured out that under healthy conditions, mitochondria transfer is predominantly from adipocytes to macrophages. But in the setting of a high-fat diet, the long-chain fatty acids actually induce release of the mitochondria from adipocytes, but they simultaneously inhibit capture from the macrophages, direct effect of the long-chain fatty acids on the macrophages. And so it's the macrophages that actually make the decision about whether or not these mitochondria are released into the circulation and get to the other tissues that are further away. So we think that actually the macrophages are, the mitochondrial transfer to macrophages actually controlling the decision about whether or not mitochondria are released and distributed to other tissues systemically. So we have a really diverse and wonderful group that we've recruited in the last couple of years. Everybody who's shown here has contributed to this manuscript and this work. Especially, I want to just call out Rachel Field and John Moley and Marina Rowan from the lab. Steve Teitelbaum was an amazing mentor who let me work on this during my postdoc and as I was starting my lab as well. And my contact information is listed on the bottom right. So if you have any questions or if we run out of time, feel free to reach out to me by email or through Twitter. Thanks so much for your attention, and would look forward to any discussions.
Thank you, John. That was really interesting. With a couple of very nice talks today. So as John just said, if you have any question, you're more than welcome to type that in the chat or just to raise your hand. So maybe I will get it started for now. So I was curious when you showed us the transcriptional changes between the macrophages that can uptake the mitochondria versus the ones that cannot uptake the mitochondria. You just briefly told us that there were some changes both at the metabolic and transcriptional level. I was wondering if you could give us more details about that.
Yeah, so I can tell you some. So there are many genes that are differentially expressed between these two macrophage subpopulations. Some of the ones that are particularly intriguing to me are that the HIF1 alpha tissue factor pathway, those genes tend to be enriched in the macrophages that have taken up mitochondria from adipocytes. They tend to have a de-enrichment of some of the aerobic respiration associated genes, which kind of fits with some of the existing literature suggesting that this process supports aerobic respiration of the recipient cell. And then also many of the collagen synthesis genes are differentially expressed. But we actually published the entire analyzed data set, so you can go take a look and find your gene of interest.
Thank you. So I will get started with the questions now since we have a few minutes left. So by Luca De Feo, he says, great talk. Does X1 deletion in macrophages affect their immunomodulatory function, skewing them towards M1 or M2 phenotype? Do transfer mitochondria fuse with the one inside the recipient cells? Thank you.
So the first question, I don't know the answer completely. It looks like it's not so simple as M1 versus M2 polarization. It looks like the mitochondrial transfer seems to be associated with a number of changes that don't really fit that paradigm. So I'm not sure I can answer the question very cleanly yet. The question about fusion and fission, that does happen. Some of these mitochondria are actually getting into the macrophage. We can see them undergoing fusion and fission with the mitochondrial network. And there's actually even some data from other labs suggesting that when mitochondria are captured, they can somehow escape the endolysosome, much like some bacteria and viruses can do. And they can, so there's a thought in the literature that I'm very intrigued by, which is that they can actually get incorporated into the mitochondrial network of the recipient cell. Some of them get degraded, and some of them also seem to be transported and exported on the other side of the cell. So I think there are probably at least 3 fates for these transferred mitochondria.
All right, okay. So I think this also answers one of the other questions that we had here in the chat. I will just ask one more question since it's 6 p.m., but feel free to reach out to John if you want to ask your questions directly. So just like a very quick question, do you know if macrophages take up anything else from adipocytes or just mitochondria?
I think it's possible that they're sampling other organelles too. It's also possible that it's been shown that they can also capture some little lipid droplets that are released from the adipocytes. So yes, I do think that there is probably other material that can be transferred from adipocytes to macrophages. It's not just mitochondria.
Okay. So I'm seeing a question here from the Open Science team. We're allowed to go a couple of minutes further. So if you're happy to answer a couple of more questions.
Yeah,
okay.
Happy to.
Yeah, so just go in order then. Let me just see. So Eugene is asking, could it be that enhanced mitochondrial transfer to macrophages detrimental? So I didn't just read again. Could it be an enhanced mitochondria transfer to macrophages detrimental? So I'm guessing what happens if they transfer too many mitochondria, if I'm reading this question correctly.
So I'm not aware of any data on that yet. There is some work that is coming out soon that suggests that like what we found in the white adipose tissue, that mitochondrial transfer seems to be metabolically beneficial. And if you disrupt that process, that has metabolic deleterious consequences. I'm not aware of any studies yet suggesting that enhanced mitochondrial transfer, it compromises metabolism. I suppose it is possible that this could lead to depletion of mitochondria from the adipocytes over time, and that this export of adipocyte-derived mitochondria might contribute to the gradual decline in mitochondrial biomass in the adipose tissue. So it is possible, but I just don't know of any data yet.
Okay, great. And then we have a question from Miriam as well. So I don't know if you want to unmute yourself and just ask the question directly. Cool.
Awesome talk, John. I have a question about what you said about these energy expense. So that these knockout mice, well, where the macrophages are knockout, they are more efficient or they use less energy. You said that this would be beneficial. But my question is, where would this be beneficial? As I think high-fat diets are not, they're pretty recent, or is that my mistake?
Yes, so I have to be careful how I answer because it's not all my data, but I think that what is happening is that this sort of riffing on the very nice cell paper from the Hidalgo lab, Andreas Hidalgo's lab, showing that macrophages are able to degrade damaged mitochondria from cardiomyocytes in the tissue. This seems to be also a pathway involved in adipose tissues, and that they can preserve the metabolic homeostasis of the adipose. So I think that that's part of the answer. But I can't really say a whole lot more to help, but stay tuned. There's a very nice paper coming out very soon that is on that very topic. Thanks.
Cool. Edgar is asking, how long can the transfer mitochondria survive into the recipient cells?
That's a good question. I don't know yet. And I don't have an answer, unfortunately. I wish I had an answer to that question.
Yeah, it's a lot of very interesting questions. So I guess we can have one last question now. So again, great talk. I feel like this is the current theme for both of you. All your questions has with great and amazing talk. So great job for you both. I think this one is more mechanistic. So is heparan sulfate specific for mitochondrial binding? Do you know what is the ligand on the mitochondrial membrane?
We do not know what the ligand is yet. I'd really like to know what it is. I will say that heparan sulfates are sort of misunderstood as being non-specific interactors. They actually have a very complex post-synthesis modification system involving not only sulfation, but also epimerization. And so you can get, and it can be very variable in length. And so you can get these 3D conformations on the exterior of the cell that are actually like very specific receptors for various factors, but we don't have good tools to characterize and study those features. So they all get lumped together in one category. I think there is specificity that's conferred from heparan sulfates because of this. And the best data that we have so far is that heparan sulfates are not involved in all phagocytic processes. They seem to be required for optimal capture of mitochondria by macrophages, but they don't impact capture of latex beads at all. but we can increase phagocytosis of latex beads in macrophages, and those stimuli actually impair mitochondrial capture. So I think that there are different pathways. There's got to be some specificity to it yet, and I just don't know what it is.
Okay. Okay, great. There's actually one question that popped up just after I said that was the last question. So if you don't mind, just this last one, then I guess we can just close it here. So from Brian Campos Salazar, he says, fantastic data, John. What would be some mechanism behind the reduction in mitotransfer to macrophages by LFCAs? Any hit maybe you found in your CRISPR screen that could suggest that?
Yeah, so I don't want to spill the beans too much because it's not published yet, but we thought initially it would be CD36, which is a scavenging receptor that binds fatty acids, and it has been shown to signal to macrophages and induce an inflammatory response. And it turns out it's totally independent of CD36. So we went back to the CRISPR screen, and I don't know how yet, but it looks like these long-chain fatty acids actually reduce mitochondrial capture by macrophages, and it's completely dependent on heparan sulfates. So somehow, I guess the roads all turn back to exit 1, and that's, I think, the exit ramp for us. But yes, it does seem like heparan sulfates are somehow mediating the impact of long-chain fatty acids on mitochondrial capture. Release from adipocytes, that's another question, and I don't know that.
All right, great. Okay, since we're 7 minutes past the hour, I guess we can finish here. Thank you very much to both our speakers today. And thank you very much for the Open Box Science team for inviting them and me as well. I don't know if they want to say anything at the end of the session, but if that's not the case, then I guess we can finish here.